Sorry. Now let's talk about the biology of these oak apples. Oak apple galls and galls in general are a common strategy for many invertebrates, forcing its plant host to create shelter, defense, and food by manipulating the plant's developmental pathways. Surprisingly, little is known about the developmental pathways of oak apple galls, and there can be a lot of variation in pathways between species. However, the most prominent idea is that the wasps secrete enzymes so that the larvae can establish themselves and then induce phytohormone transportation or production to form the gall itself. When a female wasp lays her eggs in the host tree, she secretes along with them venom and ovarian fluids. Protein components in the wasp venom may help degrade plant cells, allowing the larva to establish itself within the plant. Other enzymes may play a role in suppression of plant defenses, induction of other plant responses including production of sugars, proteins, and lipids, and synthesis of defensive compounds against pathogens such as bacteria and fungi.
Gross! That sounds like these wasps are throwing up enzymes all over the place. They are probably anxious about their babies finding a new home, but the female wasp has no reason to worry. The friendly tree is going to help this little insect build its home. The larvae and tree both work together, and this is where uncertainty lies. The molecular mechanism of insect induction of gall formation is still unknown to scientists, but many have hypotheses. Gall growth is going to begin at different times for certain species. Some species start before the egg hatches, and some they grow when the larva is still small. In order to initiate growth, there needs to be some stored meristematic tissue. There are studies that have found that gall-inducing insects can convert source tissue into meristematic tissue by expressing class 1 NOx gene. Some studies find that class 1 NOx gene has a positive regulatory impact on plant hormones such as auxin and cytokinin. It is also seen to negatively regulate gibberellins by activating GA2 oxidase. In our class, our wonderful instructors have taught us all about these hormones. Auxin is known for cell elongation and division, phototropism, lateral root initiation, vascular differentiation, and root and shoot development. Cytokinins are known as repressors of senescence. I could definitely use that for my fruits. And it also impacts cell division, shoot initiation, apical dominance, and shoot and root development. And the last hormone I want to mention are gibberellins. Gibberellins promote cell elongation and division, as well as seed germination, flowering, and sex determination. Wow, I feel like a plant biologist already. Thanks, instructors, for this knowledge. Now, back to our conversation about meristematic tissue. Meristematic and parenchyma tissues are very important in gall formation in some species. Once the host tissue, such as a stem, leaf, or flower, is de-differentiated back into meristematic tissue, the normal expression of this past tissue characteristics are gone. There is subsequent re-differentiation into specialized tissue. For instance, here's an image of a centipede larva within a gall. By secreting enzymes and plant-inducing hormones, and with the help of class 1 NOx gene, a new layer of tissue is formed. This tissue is usually lined with nutritive tissue called parenchymal tissue. This is what the larva is believed to feed upon. Parenchymal tissue accumulates both macronutrients, such as nitrogen and phosphorus, and micronutrients, such as iron and manganese. In other words, the larva creates a nutritive sink for itself. As the larva continues to grow and continues to eat the layer surrounding it, neighboring vacuole parenchymal tissue is to be converted into the more nutritive tissue, and this process will continue until it has eaten through the entire parenchymal layer and up until it hits the sclerenchymatous layer of the gall. This tissue is lignified, and our larval friends do not like to eat this stuff. Yuck.
Now, we'll explore the world of Oak Valley wasps, examining the life cycle and the relationship these insects have with oak trees. It begins with egg laying. The female gall wasp embarks on her mission, carefully selecting specific parts of the oak tree to lay her eggs, whether that be twigs, buds, or leaves. As the eggs hatch, the larvae stage commences. The larvae secrete chemicals, compelling the oak tree to form a protective gall around them. This gall is not just a sanctuary. It provides nutrition and safety from predators and the elements. Inside this natural fortress, the wasp larvae enters the pupal stage. Here, in the heart of the gall, a transformation occurs, the metamorphosis from larva to adult. They undergo the complete metamorphosis from larva to adult in just a few weeks inside these smaller galls. Finally, the adult wasps emerge, ready to continue their cycle. But what does this mean for the oak trees, these unwilling participants in the gall wasp's life cycle? Gall wasps are entirely dependent on oak trees for the life cycle. While it's true that the formation of galls diverts resources from the tree, healthy oaks are generally not harmed by their presence. This resilience is the result of millions of years of coevolution, where oak trees and gall-forming wasps have developed a balanced relationship.